Okay, good afternoon, everybody. Thanks for being back and being punctual. Hope you all had a good lunch break. So we have continuing with an exciting program for this afternoon. But before we get started, just a couple of announcements. We were supposed to go out on a boat ride, some of us tonight, but unfortunately, due to the weather, that's been canceled. We'll have to see whether we'll be able to, or some of us might be able to go out tomorrow, but we'll wait and see about the weather. Second announcement is, as many of you probably saw last night, part of the organizing committee has been so kind to bring out some nice kegs that we can enjoy for the evening. So if you're interested in making a donation for that, please see Katie. So with that, let's jump into this afternoon. So what we have lined up for this afternoon is that we'll start with 3 additional working group presentations to hear about some of the great achievements that have been made. And then after that, we will have a coffee break. Then following on Katie's initial presentation last night, she'll tell us a little bit more about how this network came together, and we'll have a group discussion about the future of the network. And then we'll have another panel discussion that'll be focused on meta-analysis. So let's get started. The first working group presentation is by Rachel Toxidlowski, who'll talk about predicting diversity and divergence in the sea, and she's on Zoom.
Yeah, so can I go ahead and share? Okay, and you can hear me, I assume? Perfect, okay. Let me get set up here quickly. Okay, can you see a title slide?
Go ahead.
Yep, okay, perfect. So thanks for that introduction and for letting me tune in virtually today. I'm really excited to share what our working group has been up to over the past few years. So just to introduce myself a little bit further quickly, I'm currently a postdoc in Gideon Bradford's lab at Michigan State University, and this has been the main project that I've been working on for the duration of my postdoc. So I was actually moving to Michigan when you all were there for the very first RCN meeting when this was initially dreamt up. And my expertise is really centered on understanding relationships between ecological processes and evolutionary outcomes, more specifically the determinants and consequences of genetic diversity in wild populations. And I'm really excited to announce that last week, I officially accepted a permanent research position with the United States Forest Service. And I'm going to be developing and running a population genetics research program with them, as well as a sister center to the National Genomics Center. And so I'm mentioning this quickly because I'm going to be looking for new collaborations. And so if you're interested, please don't hesitate to reach out. I'd be delighted to chat as I'm working on building these new programs. Okay, so back to the RCN project. Our working group essentially wanted to identify patterns and potential drivers of genetic diversity and divergence in the sea. So today I want to go over the power of macrogenetic studies, some tools for macrogenetic work that we've developed, aka how we're improving on previous studies, aka we bit off a lot with this project, aka why we're still not done yet, and then end on some preliminary results. So I suspect that at least some of you are familiar with the macrogenetic framework, but just to get us all on the same page and kind of get the brain juices flowing, answering some of the biggest remaining questions in evolutionary biology fundamentally relies on estimates of genetic diversity. And so some of these questions include things like, why are there more species near the equator than near the poles? Why does genetic diversity often fall into this surprisingly narrow range compared to the range that we would expect based on the variation in population sizes? Why are there more individuals often near the center of the range than near the range edges? And then finally, what determines those species range limits themselves? And in addition, as I suspect we all appreciate, genetic diversity is also hugely important for conservation in the Anthropocene. So getting a good handle on species level genetic diversity in wild populations and what predicts that is a really crucial step both for being able to understand or advance fundamental ecological and evolutionary research and for informing conservation strategies. And for the first time, the data are actually available to be able to investigate these questions at the genomic level and in a sufficiently powered way. And this is really primarily a result of the development of next generation sequencing technology. So we, being kind of the grand we here, are currently generating genomic scale data, sequence data for wild individuals at an exponential rate. And this is really fueling a wave of exploration into the determinants of genetic diversity. The real power of these studies then is that they allow us to connect ecological processes to evolutionary outcomes. So in kind of classic empirical genetic studies, they're often focused on connecting genetic diversity to evolutionary dynamics for one or a few species at a time. And this is because measuring things like individual phenotypes, genotypes, and fitness is often very time intensive and can cost a lot of money as well. But working one species at a time like this, as these intensive approaches often require, doesn't really give us much insight into how ecological processes or traits are mediating these evolutionary forces. And this is really because each species only has an N of one or a few in this blue outer ring. But if we combine hundreds of these empirical data sets across many different species and many different environments, we can start to understand how biotic and abiotic predictors are mediating evolutionary forces and shaping species level genetic diversity. And then if we think about taking this one step further, if we can identify common patterns that are connecting these green and yellow rings to the blue outer ring, we can in theory work in reverse for species where we know features in the blue ring, but lack genetic data for them. And this could prove really valuable in making conservation and management decisions, in addition to gaining insights into some of those fundamental questions that I started with. So our working group wanted to apply this macrogenetic framework to marine systems, which, as is often the case so far, have received less attention than terrestrial systems in this macrogenetic context. And specifically, we wanted to know, how is genetic diversity structured within the sea across a broad suite of species? And then to what extent do biotic and abiotic traits explain species level genetic diversity? And kind of embedded into these questions is the longstanding question of what generates population structure in the ocean. In addition to focusing on marine species, we wanted to improve upon previous macrogenetic studies in a few key ways. So those included including more individuals. So most studies, or many macrogenetic studies previously, have only used one or a few individuals to represent each species. We also wanted to use genomic level data, again, in contrast to the one or few markers that had previously been used. We wanted to standardize bioinformatics steps, and then also account for heterogeneity in spatial sampling. And I'll dig into what those mean a little bit more. And then we wanted to perform all of our final analyses in a phylogenetic context. And all of these steps should really lead to more accurate estimates of genetic diversity, and thus a more accurate picture of the traits that are or are not predictive of said diversity. So this brings me to the tools part of the talk, aka what we thought we were getting into versus what we actually were getting into. And I think that this is a pretty common theme that we can all relate to in research endeavors. But in this case, I wanted to take this opportunity to step through some of the tools that we've developed, because I think that we've really done a lot of important legwork that can be used to push other macrogenetic studies forward, as well as individual studies. And then also, since RCNs are really about fostering collaborations, I wanted to be able to highlight some of the numerous fruitful side projects that have developed to support this core research effort. I also want to take a brief moment to give a sincere shout out to the international dream team that I've been co-leading. With Gideon, we really have relied on the full breadth of expertise on this team, which ranges from population genetics to marine biology and deep natural history, physical oceanography, bioinformatics and large data, and theoretical modeling. So the first thing that we needed to answer our research questions was georeference sequence data, which brings us to tool number 1, which is about improving genomic metadata. So like I mentioned earlier, genomic data sets are being generated at an exponential rate right now for wild organisms. However, when I started exploring these data sets in NCBI, which is kind of the big international repository where genetic data live, only about 15% of the potentially usable data sets there had latitudes and longitudes associated with them. So as a result, Erik and I decided to co-lead what we called a remote datathon during summer #1 of the pandemic, where we essentially paid graduate students who couldn't work on their own research as a result of the pandemic to hunt down latitude and longitudes and other missing metadata in associated published papers. So these data were sometimes available, but not linked to the genetic data in the repository directly. And as a result, we were able to resurrect over 12,000 individual samples to use in DivDiv, which is what we've nicknamed our project or our working group. And while we were at it, we also resurrected spatial coordinates for an additional almost 30,000 samples, which we're now working on uploading to Geom. Geom is just a repository for storing genetic metadata or genomic metadata. And so these samples will now be able to be used in future studies, macrogenetic studies, and also for genetic diversity surveillance. We also published a paper where we drew attention to this lack of genomic metadata issue and pushed for improvements. And to our excitement, and I think quite a bit of shock, about 3 months later, the International Genetic Repository announced that they're actually going to start requiring minimum spatial temporal metadata to be deposited with all newly uploaded sequences. So this was a pretty exciting outcome that I think should both improve our ability to conduct future macrogenetic studies and research, and our ability to monitor genetic diversity in wild populations. We were able to obtain genomic data sets for 118 different marine species, and you can see we're looking at a subset of them here, and you can see that we have quite the range of taxonomic diversity. Here are all 118 of those species mapped onto a phylogenetic tree, and you can see that we have some brown algae, angiosperms. Here are a bunch of the corals. We have some marine mammals, a lot of bony fishes, crustaceans, mollusks, etc. And in addition to covering much of the tree of life, these species or these data sets also cover much of the globe. So we've accomplished our aims of using multiple individuals per data set that are georeferenced, sequences that are genomic in scale, and that encompass a broad taxonomic diversity. So this brings us to our next aim and tool #2, which is focused on standardizing the bioinformatic handling of the genetic sequence data. So most studies are using, or most previous macrogenetic studies have used the published values for genetic diversity in the study. So they're just pulling those values out of the reported, like out of the results section. But those values are often relative to that data set or to that species. And we were also concerned about differences in bioinformatic choices that had been made between these different data sets. So we decided to start all the way back with the raw reads and designed and built pipelines that can automatically download raw sequence reads of interest from the sequence repository, clean, filter, and trim them in a standardized way, and then assemble them. In this case, we chose to use the program stacks, assemble them using many different combinations of assembly parameters, and pick the optimal assembly parameter for each data set, or set of parameters for each data set. And we were able to accomplish this by making these pipelines highly parallel and then running them on high throughput computing clusters. So here we're looking at the stacks portion of the pipeline, and this specific pipeline cuts computing time from hundreds of years to days or weeks, again, by making this pipeline highly parallel. And then recently, Derek Moore, who is a cloud computing architect at Penn State, has been working on translating this code so that it will also run on a public cloud. So we are running these on academic clusters for our own analyses. And so now we're working on publishing all of these different flavors of the workflows for others to use in their own research. Now that we've assembled hundreds of genomic data sets in this standardized way, we want to use these data sets to estimate species level genetic diversity, which brings us to the third and final tool that I wanted to highlight today. So in macrogenetic studies, essentially we want to treat microevolutionary parameters as species level traits and then analyze them in a macroevolutionary context. And when we do this, we need to think really carefully, or we should think really carefully about the number that we're using to represent species level genetic diversity. And this is because these estimates of genetic diversity are sensitive to sampling schemes. So imagine that each of these kind of shapes here represents a species range, The circles inside them represent individuals and the colors represent different alleles. And we care about estimating or generating comparable estimates of species level genetic diversity across these 5 different species. So even if we sample each of these species identically, you can see that we're capturing species-wide diversity in some species, but not in others. And the reality is that the proportion of the species range that is sampled and the spatial distribution of genetic diversity across that range are both often variable. So we can see the sensitivity to sampling schemes within real empirical data sets. Here I've just pulled one of the clownfish data sets out to illustrate this for you. And you can see that we get different estimates of genetic diversity if we sample different numbers of individuals, if those individuals are distributed randomly versus clustered tightly together in geographic space, and individuals from a small portion of the species range compared to across the entire geographic extent of that range. So when we estimate species level genetic diversity, we should be using an approach that's aware of the geography of the sampling and how that relates to the geography of the range. And this is essentially what we've been working on most recently. Zach Hancock, another postdoc in the Bradford lab, has been leading this effort, and Gideon and I have been playing support. And the idea here is basically that this isolation by distance curve, or kind of how genetic diversity and divergence are accruing over geographic space, includes information in it about migration rates, population size, and other population genetic quantities that we might be interested in. So that is to say, if we can reconstruct this curve, if we know quantities like population size and migration rates. So we've developed a Bayesian model based on a classic Wright-Mallico formulation that estimates the neighborhood size, the species level genetic diversity, and the migration rate that best reproduce a given empirical isolation by distance curve. So in this case here, we gave the model pairwise geographic distances and genetic divergence between all the pairs of clownfish in that previous data set that we were just looking at. And then we're getting out an estimate of species level genetic diversity. And so now we're working on testing the model performance and accuracy using simulated genetic data. And so far we're doing a pretty good job, which we're keyed about. So here you can see we're looking at estimates of neighborhood size for simulated data across a variety of dispersal abilities and different population densities. And 0 here means that the parameters that we're estimating are hitting the true parameters. So this brings us back to our final or our original core research aim now. And take these with a grain of salt because we're still confirming model behavior, like I just mentioned. But these are the empirical estimates that we have so far for neighborhood size and genetic diversity across about half of the species in our data set. And we're seeing considerable variation across these species, which is exciting because it should provide good power for testing the extent to which our chosen traits influence or not genetic diversity. And I just want to reiterate once more that few studies to date, especially in this macrogenetic context, have used spatially explicit rarefaction approaches to estimate species level genetic diversity. And so as a result, we expect that many current species level estimates of genetic diversity are actually underestimates. And importantly, these biases are not expected to be uniform because they should be more pronounced in species that have stronger population genetic or spatial genetic structure. At the same time that we've been working on the PopGen side of things, our sub-team, lovingly known as the Mera-Nerds, has been combing the literature and online databases to aggregate biotic trait values. Most of these, perhaps not surprisingly, have to do with population size, demography, and/or dispersal. And then our sub-team named the Oceanographers has been calculating abiotic predictors, and those are mainly focused on metrics of range size and environmental complexity across that range. And so now we're finally at a point where we can start to put it all together to understand how these traits relate to levels of genetic diversity. So to do this, we're using a Bayesian framework, and we're controlling for phylogenetic relatedness between the species. And so here we're looking at the effect sizes and significance for a bunch of those predictor traits and how well they predict species level genetic diversity and neighborhood size. So again, take these with a grain of salt, but so far we're seeing quite a bit of action in neighborhood size, but less so in how predictive these traits are of overall levels of species genetic diversity. Finally, I've just pulled out a few examples of what these relationships actually look like. So over here on the left, we're looking at the effect of spawning mode on neighborhood size. And this relationship is currently in the direction that we hypothesized, which is essentially that higher dispersal should lead to larger neighborhood sizes. And then on the right here, we're looking at the effect of larval feeding on genetic diversity. And this relationship, there are some disagreements in the group about which way we expected it to go. But the working hypothesis for this current direction that we're seeing is that planktotrophic species produce a lot more offspring. So perhaps they have higher mutational loads, and they also might be less susceptible to population bottlenecks if they have larger population sizes. So these are the types of relationships now that we're working on testing. And in the end, we'll have produced robust estimates of marine genetic diversity. We'll have tested biotic and abiotic drivers of genetic diversity. And along the way, we've developed some key tools to improve macrogenetic studies. And these include improving genomic metadata standards, standardizing or building standardized high throughput bioinformatic pipelines, and developing the spatially explicit model for estimating population genetic quantities. And these large macrogenetic studies then will give us a chance to understand how ecological processes are mediating evolutionary forces and shaping species level genetic diversity. And they can also help us to understand fundamental questions in ecology and evolutionary biology, and to design effective conservation strategies for threatened species in the Anthropocene. So with that, I'd like to give a sincere thanks to the many people that have contributed so far to this project and to our funding sources, and I'm happy to answer any questions. Thank you.
Great. Thank you very much, Rachel. Excellent talk. Do we have any questions? Well, I have a question then. So that pipeline that you've developed to automatically download raw data, how versatile is that? Is that specifically for any one type of data, like RAD data, or could it also work with whole genome data or sequence capture data, or how is it set up?
Yeah, so the nutshell is that we always find new data sets that break it in some way, but because of that, it's becoming more and more flexible. We have designed it purposely to work with whole genome data and RAD data. But I think with minimal tweaking, you could basically download anything you wanted from NCBI and run it through this pipeline. And it would just be a matter of, basically, we've kind of pulled in, like we use CutAdapt to do the trimming, and then we design, we have a couple different flags of like how we want that trimming to work. And so by going in and just changing some of those flags for your own specific types of data, I think it would be I don't think it would take a lot of work to make it flexible for other types of data sets as well. Yeah, so, and then we've written in some algorithms, for example, that kind of look at what's the distribution of read lengths, and then pick a trim length that is going to keep like most of the reads and spit them all out where they have one common read length, because that's what Stacks requires. Not all programs require that. So there are a lot of decisions that we had to kind of come to a consensus on for this specific project, but changing individual numbers here and there in the pipeline would allow it to be really flexible. Does that speak to your question?
Oh, great, thank you. And I think we just have time for one more question. Jamie? Okay, sorry. So with the Stacks, Catherine Silverman on Zoom is saying, with the Stacks genotyping step, how does the pipeline choose the best set of parameters?
Yeah, so that's been kind of the bane of my existence. Right now, we've decided to use what's called the R80 rule, which I don't know if you're familiar or not, but it's basically you pick the set of parameters that gives you the most loci that are scored in at least 80% of all of the individuals. And that's from Julian Ketchen, and who's the developer of Stacks, and his team have come up with that rule, and it seems to perform pretty well. So this is something that this was kind of one of the biggest challenges overall of working with a large data synthesis project like this, is you have to walk this line right between doing 100 PhDs over again versus doing something that's nonsensical. And so we landed on this R80 rule, and it seems to be performing well so far. For when I'm working with my own individual data sets, I prefer to look at a lot of kind of plots and look for asymptotes in different features of the data, for example, to pick those parameters. But that's how we're doing it at this scale.
And I think we'll take Jamie's questions while René sets up for the next presentation.
Rachel, just quickly, this is Jamie. What are the units on the neighborhood size? Are those individuals or?
Yes, those are, yeah. I mean, they're not exactly individuals, but it's basically a radius. It's the number of individuals in a radius of one dispersal distance that are reproductive. So like kind of the potential mates within one dispersal distance of you. Does that answer your question?
Yeah.
Okay.
Okay, great. Thank you very much, Rachel.
Thank you.
Okay, so the next working group we're going to hear from is the presentation is going to be by René Clark, talking about the reporting from the Temporal Genomics working group.
I'm setting up over here because my computer does not play nicely with podiums, so sorry about that. But I'm Renee. I did not do a lightning talk yesterday, so I'll quickly introduce myself. I am finishing up my PhD at Rutgers University, where I do a lot of work characterizing patterns of adaptation and genetic diversity in tropical reef fish. Not going to talk about that today. Instead, I'm going to give an update on the Temporal Genomics Working Group that I help lead. Before I go into that update, I first want to give a huge shout out to all of the wonderful working group members that have put countless hours into collecting all the data I'm about to show you. So besides myself, there's Brendan Reed, Allison Sawkins, John Whalen, who are all here at the workshop with us. And then also Katrina Catalano, Kira Fitz, Erik Garcia, Kyle James, Anthony Sneed, and Malin Pinsky, who's both my PhD advisor as well as the faculty mentor for this working group. Okay, so I figured I'd start with the definition, especially given the panel we had right before lunch. I know a lot of you are already familiar with temporal genomics because a lot of you mentioned it in your lightning talks yesterday, which was super cool. But for those of you who are less familiar with this area of study, I guess, we define temporal genomics here as just any study that incorporates temporal or time series data into analyses. So it's just a study that has genomic data from 2 points or more than 2 points and is looking at change or patterns across those 2 time periods. And these types of studies are really incredibly powerful, particularly because they enable the direct characterization of genomic change. So instead of having just a contemporary time point and we're looking at what we see in that contemporary population or trying to use those contemporary data in order to project backwards to figure out previous demographic scenarios that created what we see today, we can use time series data to say we have point A and point B, and we can directly look at the change between those 2 time periods to see what's taken place. And so this figure here is from a paper in 2021 that just shows different ways that we can use temporal data to ask and answer different population genomics questions. And there has been like a great rise in temporal genomic studies in the past, I would say, especially with that next generation sequencing. And so as all of these studies have started to come out more and more, so too have there been a lot more reviews looking at how do we best do different types of temporal work. And particularly reviews looking at 2 kind of aspects of study designs. One, how do you access historical data? Like what museums are out there that have them? What repositories and databases and archives are there for you to go back and get historical data if you don't know that they exist? And then especially how do you unlock historical DNA? How do you do extractions and library prep and sequencing and what type of metrics and markers can you use. Because historical DNA, ancient DNA can be a little bit finicky if you've ever worked with it. It's a bit delicate, it's a bit difficult to work with, as DNA can be in general. And so there's a lot of reviews looking at how do we access that DNA so we can analyze it downstream. But despite all of these reviews, there's still a lot of unanswered questions and how we can design these types of studies. And so that's one of the main goals of this working group, was to try and figure out how do we best design temporal genomic studies to kind of maximize our power and best detect change if change has really occurred. And so there were a lot of different deliverables or products of this working group, and I'll mention a few towards the end. But the big goal was to conduct a systematic literature review and kind of write a paper doing two different things. One was to assess the state of the field, the current state of the field of temporal genomics. So what kind of questions are most asked and how are they asked? Like what taxa are we often doing these types of studies in? What types of systems with what types of data? And are we interested in questions of adaptation or population size and things like that? And then the other part of this review was to develop recommendations for detecting recent evolutionary change from temporal sampling. So again, how do we best do study design and sampling design to increase our analytical and statistical power? So I'm mostly going to talk about the state of the field now because that's what I have before, but I'll get to some of those recommendations at the end. And if has questions or wants to talk about recommendations at the end, I'd be more than happy to chat about that as well. So briefly, methods for how we gathered the paper and the data that went into this literature review. We did a keyword search in Web of Science for these terms here: temporal genomics, historical DNA, hdNA, aDNA, all of those terms. We got back about 40,000 papers because this is, a really big field and it spans studies from like a few days to thousands of years. So we really needed to narrow down what time and what types of studies we were going to focus on. And so the studies going into this data set are limited to studies that focus on non-domesticated or wild animals using DNA samples that have been collected in the past 200 years. So anything older than about 1800, we did not include. In this data set, we're really looking at more recent historical change from the Industrial Revolution on. And all of these studies also incorporated at least 2 time points, so no single time point studies, so had to look at change from one period to the next. And they were investigating change in questions of either adaptation, diversity, connectivity, or population size. Okay, so I'm going to go through some of the highlights of the study. We, again, collected a lot of data from all of these studies. So if you are interested in a trend or a pattern that you're not seeing here, I'm not presenting, we probably recorded data on it. Just come talk to me. I'd be happy to chat about it. But this is just a general breakdown of, again, what are the studies looking at? What are these temporal genomic studies in our data set? Where are they and how are they being conducted? So we found that of those about 40,000 studies we started with, we narrowed it all down to about 230 studies that we collected data on. And most of these studies looked at changes in genetic diversity in terrestrial systems. So we did have studies looking at change in population size, connectivity regimes, and adaptation, although relatively few studies looking at changes or looking at adaptation through time. And again, most of these were in terrestrial systems. And in general, this is a theme that's going to come up. A lot of these studies were looking at big charismatic species that had well-documented population size changes or well-documented effects from over-harvesting or habitat degradation that people wanted to go back and look at to see the genomic consequences of those changes. Another thing that we looked at was not just, what kind of genomic change are people interested in, but what's the driver of that change? Are we looking at change due to habitat loss or exploitation or invasive species or climate change? So we narrowed down the potential drivers or human causes, or not always human causes, but causes of change and processes listed here on the board. And then there's, of course, studies that didn't really have a driver of change. We're basically just like looking at population size through time. And what we found was that most of these studies, and it didn't really matter whether they were looking at adaptation or diversity or so on, most of these studies investigated change due to habitat loss and human exploitation, which again matches this kind of idea that these are well-known, like big case studies. We know elephant seals went down, so let's go back and look at elephant seals and see the genomic consequences of that bottleneck. Or this bird was over-harvested, let's go see what happened there. We also saw a lot of studies looking at changes due to just environmental variation. So this is things like we want to see if the connectivity patterns are consistent from one year to the next, or do they change because of changing currents? Does genetic diversity, is it stable through time, or does it just change randomly and fluctuate? Those types of studies were also pretty common in our data set. We found, so when I think of temporal genomics, I often think of using museum or historical samples from an archive, but a lot of studies in our data set, about half of them were what we call here kind of pre-designed studies, or they're samples that were designed by the authors or by the researchers, and they went out and collected every single year. So all of the samples in the study were collected in the field by the researchers themselves, instead of going back and kind of quickly gathering what was available, if it was available from museums, which was actually pretty interesting. I personally did not expect to see that many kind of pre-designed studies in our data set. But what you can tell from this graph is that the number of these opportunistic studies that use museum samples are increasing quite rapidly through time. And I think that is an indicator of the better techniques that we have for extracting DNA from museum samples. And so we're better able to conduct these samples as we get into 2015 and move on to 2020. And then just one last figure I'll show here. We also looked at kind of the duration over which these changes were, and did that vary on the driver of change or the system that you were looking at, or all of these different things. I'm just showing driver of change here. So we wanted to see, did studies look at change over 10 years, 20 years, 30 years, or so on average? And we found that, again, studies that primarily focused on genomic change due to habitat loss or human exploitation span the longest time frame. So looked at change over the longest period. And so these, I should back up and say, these timeframes here, so some samples, some studies collected over multiple time points. So this is just the earliest and the latest, the most recent, the most contemporary, and then the one farthest back in history, the maximum time that could have been observed. And so, yeah, habitat loss and human exploitation studies looked at change over the longest time period. And then things looking at change from a natural disaster looked at the smallest time period, which makes sense. You're looking right before and after a very quick, abrupt change. Okay, so some takeaways from these findings, and this includes that I didn't show figures for because I only have so much time here. A few things kind of stood out, and I'm kind of framing these as areas where we know relatively little that are kind of ripe for more studies. As I mentioned earlier, there's relatively few studies in the data set looking for signatures of adaptation, so relatively few temporal genomic studies looking at adaptation. And again, fewer in systems like freshwater systems or marine systems. I think the adaptation question is twofold. So I think one, I think adaptation studies are difficult, and often we want phenotypic data that we may not have available to do these types of studies. I think two, I'm not sure our search parameters actually got these studies as well as they got some of the other studies. So I do have a feeling that there are some more adaptation focused studies out there using temporal data that we don't have in our data set currently. The other thing I want to highlight is we looked at the taxa that a lot of these studies investigated. Most of the taxa were mammal, bird, or fish. Again, big, charismatic, well-studied organisms. So there's a lot fewer temporal genomic studies on insects and coral and things like that. I didn't have a good figure for this yet, but we found that most of our studies, probably unsurprisingly, looked at temporal change in North America or European, especially Western European countries. However, we did have studies that were big. That's a pretty good coverage of the globe. And if you look at it on a globe, although I don't have a figure, it does cover a lot of the globe. The caveat there is that a lot of those studies, although they have samples from Colombia, they may be samples from Colombia that are housed at the Smithsonian, or there are samples from Botswana that are housed in a museum in London. And so I do think there is a strong legacy in these studies of colonialism and a lot of questions about who has access to doing this type of work. And one thing that we want to look at a little bit more is who are the authors on these papers? Like, first of all, where exactly are the samples housed? Not just where, you know, the organisms lived. Where are the samples housed, and who are the authors and what are their affiliations, to see, again, who is actually doing this type of work. Fewer studies looked at change due to competition or disease or natural disaster. That last one especially makes sense because they're relatively rare. You have to have like the right moment and the right time to be able to look before and after a volcano explosion or something like that. Another kind of interesting factor here is that studies that looked at climate change spanned a relatively short time frame, which was surprising. So if I go back to this last slide, this is, of course, just Box and Whistler plots, but the median number of years a climate change study looked at was about 20 years, which was a lot lower than I would have expected. So that was an interesting takeaway here. And then I didn't show any data on this, but we did look at the marker and the library prep method and sequencing method for these studies and found that most of them looked at satellite markers. Fewer studies use SNPs or targeted sequence capture or whole genome sequencing, although those are increasing over time as well. And then briefly, I'll highlight some of the best practices. So a lot of these are probably pretty familiar to a lot of you and overlap a lot with contemporary if you're doing just contemporary sampling. But a few suggestions here are, of course, large sample sizes when possible. Museum sampling can make this difficult because we don't have a time machine. We can't go back and collect more fish just because we would like more fish in our sample. So some ways to deal with sample sizes, if you are interested in doing that, can be think carefully about trade-offs between sample sizes and space. Is it better to sample more individuals and sequence away with fewer individuals, depending on the type of question you're asking? You can also think about combining samples temporally or spatially. You maybe only have one sample in this year, but you have a few in the year preceding or following it. So if we have a few samples in a local area around where you're actually interested in, adding those to your sample size as well. The caveat there is you want to be careful you're not mixing populations and then artificially inflating metrics or measures of diversity, which can influence your downstream analyses as well. More time are awesome. So just before and after studies or point A and point B studies can give you a lot of information about what's happened through time, but the more time points you have, you are better able to track the trajectory or tempo of change and see exactly how things have, or have better ability to see how genomic patterns have played out through time. And the other thing here is that when you sample matters. So we know that like loss in genetic diversity doesn't happen linearly. So if you sample if you're sampling for change in genetic diversity, you want to make sure that you're sampling close enough to when, the event, the bottleneck, whatever it is that you're interested in looking at happens, because if you sample too recently, you may actually be underestimating diversity loss, for instance. So making sure that you know when you're sampling from and how it tracks with any event that you're interested in is important. And then finally, generation time matters. So I didn't show a figure for this, but when I showed you the figure looking at how we looked at number of years and over the time frame, we also looked at the number of generations that studies investigated. Because when we're thinking about evolutionary change, generation time is really the unit that matters. The more generations, the more genetic change you may expect to see, and it will at least temper your expectations for what you expect to see in contemporary samples or how things have changed. So thinking about the number of generations that have elapsed between your temporal time points or your temporal samples is really crucial when thinking about expectations from these studies. And with that, I just want to thank you all for listening. So again, I've listed all of our working, read all of those papers and pulled all of the data. Of course, a big thank you to the RCN and the workshop coordinators for funding us and for putting on this workshop so we could share our results. And I also really briefly wanted to highlight a few of the other deliverables that we put together. So in addition to this paper, which we're currently working on and hoping to submit this winter, we put on 2 different symposiums. So in the spring of 2021, we did an 8-part virtual symposium where we had a bunch of speakers, including many people in this room, come and share their work on temporal genomics. And then we had a few round panels as well. So if you're interested in any of those talks, they're recorded, they're up on YouTube. You can go back and check them out. There's a link to our website at the bottom of this that you can go to and access the YouTube channel from that website. We also did a Twitter symposium to highlight more early career scientists, again, including people in this room. So thank you to all who participated in both of those. If you want to see any of those Twitter talks, the hashtag that connects to all those Twitter talks is up on the board as well. And then we also helped put together the spring 2022 reading session. So all of that is up on the RCN website as well. So I think that was a little bit long, but thank you all. And I'll take any questions.
Thanks very much, René. Great overview. Do we have any questions, Katie?
Hi, this is Katie Lotterhos for the online people. I might have missed it, but did you show how many studies use just 2 time points as opposed to more time points? I'm just curious, like what that distribution looks like.
We did not, but I have the data to look at that for the number of time points each study had. It's a little bit difficult to go back and because not every time point was compared to every other time point, or some of the time points were only used in certain analyses. So accessing and looking at patterns in that particular part of the data is a little tricky, but it's something that we have ability and will do in the future. And then I will get back to you.
Question is, can you summarize what kind of hypotheses they're testing? Typical, what kind of results do they expect to see? I'm asking for myself, to be honest with you, because we just did this ancient moral DNA study. It works, fantastic. Now what do we do? What can we actually study?
Yeah, so I would say most of these studies, and again, I think part of this is because we limited it to in the past 200 years or so time change. So not a lot of ancient DNA. Often it was, we know there's been a serious bottleneck in this population. Let's see if we expect diversity was lost. Let's see if that actually happened. Or it was trying to, we know from public records or the history of this area that this big change has happened. Let's see if it actually had a genetic consequence. was quite often the motivation. So a lot of times people went in with the expectation there should be a genetic change. There should be some kind of decrease in genetic diversity, decrease in effective population size, change like that through time. Let's just kind of verify. We expect that happens. Let's verify that that happened and go back and look at the data. Does that answer your question? Yeah.
Are there any questions on Zoom or other?
The list of, and I was thinking about this with what you noticed, and the list of hypotheses could actually increase to make sure that you got more than just one population.
Yeah, and there definitely were like quite a few studies that said, we know there was this particular population definitely declined in size. Like we know it declined in census size, at least. This other similar population didn't. Let's see if they had similar or different changes in diversity or effective population size through time. And so I do think expanding out and adding spatial components to this, like on the one hand, let's just see if what we think we're seeing actually happen and use it from a conservation perspective. Okay, this population really, we know it's endangered and it's low in genetic diversity. Maybe that'll influence how we're going to conserve that population or treat it or species or what have you. But then I think there's a lot of studies that are just kind of testing, do bottlenecks really affect diversity the way we think they do? Or do these species really have high or low diversity the way we think they do? Or, X, Y, and Z evolutionary hypotheses that you could use in this temporal framework. And I think certainly adding spatial elements to that makes those studies more robust. Again, don't know if that really answered your question. Yeah.
I had a quick question too. So did you have a chance to look at to what extent studies had taken potential effects of DNA degradation into account and controlled for that? Because that's obviously been a huge issue in ancient DNA research, working with much samples, and obviously a few hundred years is also going to degrade DNA. But did you look into that, how studies had those issues?
We kind of stayed away from that question to stay a little, because there have been so many reviews and suggestions on how to deal with DNA degradation because it is such an issue that we kind of stayed away from those types of methodology questions in this particular review. So I say we didn't explicitly look at it, but from reading papers and reading methods of papers, I would say often a lot of, at least the papers I read, didn't really address that so much and say we tried to account for effects of degradation X, Y, and Z ways. But I don't have any good statistics on that. Yeah.
Great. Okay.
Really quickly, I could answer a bit of this question for you. Actually, DNA degradation in ancient sample is good. This is a very robust way to ascertain that what you're sequencing is actually ancient DNA and not some contamination. This is like the major criterion by which you filter your reads. Are they really super degraded ancient DNA? Yeah, so it's actually good.
Great. Thanks very much again, René. Okay, and the final working group presentation for this session will be by Katie Lotterhos, talking about meta-analysis.
Okay, so it's great to be here to talk about our working group that was thinking about how plasticity and adaptation evolve in metapopulations in nature. These are patterns known as gradient variation, and this was done in collaboration with Molly Albecker, who was our RCN postdoc, and she came into the postdoc with a strong interest in this from observing these patterns in the frogs that she worked on that adapted. These were freshwater frogs that were adapting to like saltwater marshes environments. And with Jeff Trusso, who also has a history of thinking about these problems. So this was a new kind of like new area for me to think about, and it's been really interesting. So just to give you a little bit of background, and before I start, I just want to highlight that Molly developed some of the conceptual visualizations that I'm going to show you. So she does a really great job with these kinds of data visualizations. Oh, share my screen with you guys. Sorry. Okay. I'm trying to host and talk at the same time, and it's not a good idea. All right. Let me, thanks. All right, share screen. All right. So biologists have a rich history of studying clines. Endler in 1977 wrote a whole book on the study of clines. And a cline is a measurable gradient in a character like a phenotype, but it can also be something else like a genotype or an allele frequency. And the way that we can understand the patterns that underlie these clines is to do things like reciprocal transplant experiments, where we take individuals from different populations and we put them in other locations, or common garden experiments where we take individuals from different populations and we raise them in, for example, cold environments or cold and warm environments. And by doing these kinds of experiments, we can determine if a phenotypic pattern that we see in nature is due only to the environment, only to genetics, or to some genetic by environment interaction. And it's these genetic by environment interactions that have captured the interest of evolutionary and evolutionary ecologists for really for many decades. And it's because that with these interactions, there's non-random associations between genotypic and environmental effects on phenotypes. But these are not the only way that genotype and environment can be non-randomly associated. We can also have covariance between genotypic and environmental effects on phenotypes. And I'm going to call this COVE-GE for short. And Levins in 1968 was the first to observe these patterns of covariance by studying altitude adaptation in flies. And what he did, what he noticed in nature was that body size was larger at colder environments than it was in the warmer environments. But when he brought them into the lab and raised them at common temperatures, these this pattern that he saw in nature was flipped, whereas the flies from the colder environments were actually smaller than the flies from the warmer environments. And he argued that this pattern was adaptive because large body size reduced desiccation in the dry lowlands. So there was some environmental component causing smaller body sizes at warmer temperatures, but then there was this genotypic evolution to evolve larger body sizes due to the selection at the lowland warm environments. So Levins saw these patterns in 1968. Here's just a few other classic studies that have noticed these patterns. Bervin in 1973 in frog adaptation along an altitudinal gradient, and Conover and Present in the kind of classically known Silverside example in the 1990s. But the interesting thing about all these historical studies is that nobody's actually measured this variation. And Levins in 1968, I forgot to mention, called this counter gradient variation. That's the pattern shown on the right, the pattern that the flies showed. We can also have co-gradient variation. In co-gradient variation on the left, you'll notice that as we move from environment 1 to environment 2, the genotype from green to blue is an increase in the phenotypic effect. So the genotypic effect is positive, and the environmental effect, which is the measurable phenotype from moving from environment 1 to environment 2, you might think of it as body size, is also an increase. So co-gradient variation is a positive covariance between genotype and environment. And when we go out and measure these populations in nature, we would see a very steep phenotypic climb. Countergradient variation is the opposite. So in this case, the phenotypic effect of the genotype from the second environment is a decrease in the phenotype or body size. And yet the environmental effect is an increase. So this is that slope here. So in this case, the genotypic and environmental effects oppose each other. This is known as counter gradient variation. And if we go out and measure these populations in nature, we might not see any kind of phenotypic decline across an environmental gradient. And Conover and Present really highlighted the importance of this to evolution, because if we're going out and looking for declines and we don't see them, we might think that there's not anything interesting happening at a meta-population level, even though there is. And over the last several decades, these concepts of co-gradient variation and counter-gradient variation have been ecologically and evolutionary significant, from everything from body size declines across latitudes to thermal adaptation and community ecology. And you can't really see it underneath all these pop-ups, but there's some really great reviews written on this on the bottom. And I like how the Urban 2020 paper kind of talks about co-gradient variation as evolution amplifying an ecological pattern. And in the case of counter-gradient variation on the right, evolution is dampening an ecological pattern. And these can have cascading ecological effects in space, depending on what kind of covariance we see. But nevertheless, in the last 50 years of studying these patterns, there hasn't been a quantitative method to really quantify this and test for significance of these patterns. And this visual detection has limits. We cannot test for statistical significance, and we cannot test for broader patterns of counter-gradient and co-gradient variation in nature and conduct the kinds of meta-analyses that the other working groups have been doing. So we had to start just by thinking about, how do we measure this? If our goal is to really look at a meta-analysis of these patterns in nature, how are we going to do that in a common framework across studies? And in 1989, Falconer wrote a textbook that is really very thorough textbook. And he talked about how phenotypic variance can be broken up into its component parts: environmental variance in the phenotype due to the environment, due to the genotype, due to the genetic by environment interactions, and due to this covariance between genotype and environment. But since 1989, no one has actually implemented this variance component approach to measure this COVE-GE. And so we were thinking, well, if we can't estimate a variance component, maybe we can estimate the effect size of the COVE-GE in the same way that we do a correlation, for example. So I'm going to walk you through the intuition of the sampling metric that we developed. First, we have some kind of common garden or reciprocal transplant data with genotypes raised in the environments that they came from, or that the other genotypes came from. And we measure the phenotype of each genotype. These are the genotypic effects on average across all the environments. And I'm just going to replot those on the right graph now so that you can see where this covariance comes from. Then from this reciprocal transplant data, we can measure the environmental effects. That's the average phenotype across all genotypes in each environment. So that's the E lines on that graph. And now we can plot that genotypic effect against that environmental effect for the same population. And the covariance is essentially this covariance between the genotypic and the environmental effects. And this is the sampling estimate that we developed to measure this covariance. You'll see that it looks very similar. If you're familiar with statistics, it looks very similar to a correlation, but with some very important differences. And what this measure gives you, just like a correlation, is a number between -1 and +1, where a negative number is negative covariance or counter gradient variation, and a positive number is positive covariance or is co-gradient variation. So this part of the equation is those genotypic effects that I was telling you about. This blue box indicates the environmental effects that we measure from the data. This I is an indicator variable that indicates that we only want to count terms towards the sum when those genotypes match with the environments that they came from. So when they match, we count them towards the sum. When they don't match, we don't count them towards the sum. And you'll notice that the standardization here is really different. And this was the key insight that we had, was that these genotypic and environmental effects are not independent. And so we standardize by one or the other, whichever is greater. And I say that in 2 minutes, but it literally took us 2 years to figure that out. And a lot of simulation work to figure out why the standardization wasn't simpler than that. So these genotypic environmental effects are not known exactly, they're known with error. So we use bootstrapping to develop 95% confidence intervals, and we use permutation to develop a p-value to test the null hypothesis that COVE-GE equals 0. So to test, to validate the statistic, Molly created 30,000 plus simulated patterns that we might see from reciprocal transplant experiments in nature. And I'm just going to use the example of oysters adapting to salinity because that's something that I'm thinking about. So we have a freshwater ecotype and a saltwater ecotype. And we do a reciprocal transplant. So we collect them and we put them in freshwater and saltwater environments. And we might be able to do that for maybe across 4 different salinity variables. We might get a pattern that looks like this in our data. So the metric gives you an estimate, it gives you a confidence interval, and it gives you a p-value. So this is positive. So this is an example of co-gradient variation. And we also added genetic by environment interactions in the data. So we could measure how these COVE-GE metrics fare in the presence of these genetic by environment interactions. And I think the important take-home message is you might not visually see that this pattern on the right has a strong COVE-GE pattern by looking at it. And that's what the history of this field has done, is base it on visual patterns, and this visual detection has limits. So now with a quantitative metric, we can detect it in cases that's not is obvious. So in addition, we did paired common garden experiments. So it might not be easy, like in marine systems, to go out and have like 4 different sites across an environmental gradient and collect from all those sites and put everything across all those sites. It might be easier to just go out and collect high and low salinity oysters from different estuaries across the coast and bring them in and raise them in low and high salinity environments. And this kind of design, we determined in our simulations, actually has a lot of power if you use multiple genotypes. Sorry, I forgot to use my animation there. So what kind of sample sizes are needed to detect this COVE-GE with our metric? We found that if you only have 2 genotypes in 2 environments, this is a lower power scenario. So these kinds of designs, which are the ones that have historically been used the most, have the lowest power. If you can raise multiple genotypes and you have this kind of paired design, and you can raise them, even if you just raise them in 2 environments, this kind of design has much higher power to detect COVE-GE in the data. And we also found with our simulations, a lot of things depend on power, but in general, sample sizes greater than 200 individuals across all of the samples that you collect were necessary to have higher power. So then we applied the framework to some empirical data sets. We applied it to this data set from Molly on her frogs that I was telling you about that were freshwater frogs adapting to saltwater environments. And so we detected significant counter gradient variation in her data. And we also applied it to some of Jeff's data, who he had found counter gradient variation patterns in these Littorina snails that were adapting to low flow and high flow environments. And the phenotype that was measured here was shell mass growth. And as you can see, the COVE estimate is negative, but the confidence intervals overlap 0 and the p-value is not significant. And that's, we think, is more due to the fact that this is one of those 2x2 designs that have lower power that I was just telling you about. So now we can do a meta-analysis and we can ask, how common is COVE in nature? And there's already been a few studies that have asked this question and tried to come to some conclusions based on looking at patterns in the data. The first was in 2009 by Countover. He concluded that counter gradient was more common. A second in 2020 by Urban et al., again, just looking at patterns, also concluded that counter gradient was more common. Another recent one in 2020 used a quantitative framework. The framework they used had a lot of limitations I'm not going to get into, but they concluded that the cogradient was more common. So we don't have consensus in the literature about what is common. So to get at this, Molly and Thais, who is a technician in my lab at the time, did a meta-analysis, and Molly developed some R code that hopefully she can talk about in the panel to reproduce this meta-analysis based on specific search terms in the literature. And we, from that and some additional criteria, discovered a few hundred traits across dozens of studies. So what I'm going to do is plot for you our COVE metric on the x-axis against a standardized metric of genetic by environment interactions on the y-axis. So we can ask, you know, what is more common? Are genetic by environment interactions more common? Is COVE more common? Are there specific traits that we see showing one pattern versus the other? So I want you to think about this parameter space for a minute and think about, like, what do you think we found? Do you think, if you think we found that G by E was common and COVE was rare, then we would find points in this area of the space. If you think that, you know, G by E is rare and counter gradient variation is common, we would see points in the lower left. Or if G by E was rare and co-gradient variation was common, we would see points in the lower right. So now I wish I had that poll everywhere where, like, everyone could click on the graph and I could see what you're all thinking. But what we actually saw was just patterns all over the parameter space. We found that the effect size for counter gradient variation on average was really similar to co-gradient variation on average. We didn't find any associations in the data with specific traits or endotherms versus ectotherms, but we did see that co-gradient and counter gradient variation is at least as common as genetic by environment interactions in nature, and that a lot of species show both. And that's the green diamonds in this graph. So cryptic counter gradient patterns are as common as co-gradient patterns with similar effect sizes. Patterns were not associated with types of traits, and gradient variation is at least as common as genetic by environment interactions in nature. So I think this brings us to, you know, what are the important future directions for this, for thinking about these associations and what they mean for species responses to climate change. And a lot of us have been trained to think about local adaptation underlying, or genetic by environment interactions underlying local adaptation. And one question we get a lot is, well, how does counter gradient or co-gradient variation lead to local adaptation? There's not an interaction there. I don't get it. And so it seems like a paradox. It was definitely a paradox to one of our reviewers on the paper. But the way that we resolve this paradox is by thinking about the relationship between phenotypes and fitness. And so, you know, we can have a relationship between a phenotype like body size and an environment like temperature or growing season, and that shows counter gradient variation. But the relationship between fitness in that environment could show a genetic by environment interaction. And so the key insight here is that the y-axes are actually different things that people are measuring, even though they're not always talking about them differently. So just to illustrate how this paradox arises, let's say we have silver sides that we're raising in the north, and we find that the northern genotypes are larger than the southern genotypes, and they have, and this is the relationship between body size and fitness. So larger body size has higher fitness because they have to grow really fast before the winter so they can get through the winter. Whereas if they're too small, they don't survive the winter. So they would have this kind of fitness. And now I'm just going to plot fitness against the environment here on the bottom. So if we raise these genotypes now in a warmer environment in the south, we might find that the larger genotypes grow too fast. Their metabolic rate is too high for that warm environment. They can't keep up, and so they have lower survival, whereas the slower growing genotypes have an optimal growth rate. And so these genotypes have higher fitness than the green ones. And if we just translate that down, they end up down here in the graph. And this is where that genetic by environment interaction comes from if I now combine those last two graphs that I showed you. So the first knowledge gap is like, why is there no paradox? What are these relationships among environments, phenotypes, genotypes? How is this plasticity created? Is it epigenetic? And then how does that translate into fitness? The second knowledge gap is, how does this counter gradient and co-gradient variation evolve? What kind of genetic architectures underlie this problem? And I've started doing some like theory and simulations to try to answer this. And this theory can be used to create explicit predictions that we can test. So if you have data sets that you think would be good to test some of these predictions, come talk to me while we're here, and maybe we'll figure out a way to test this. And then the third knowledge gap is, how do co-gradient or counter-gradient patterns alter population trajectories to climate change? And there's been some different verbal arguments about this, and I'm not really clear at like whether or not the things that people are verbally predicting would be true. So for example, in co-gradient variation, we have warmer genotypes having higher growth rates than colder genotypes. But under climate change, it's going to take that northern population warmer. It's going to take that southern population warmer. What does it mean for these colder genotypes to have a slower growth rate that's increasing with lots of capacity here? Whereas these warmer genotypes, maybe they're really close to the edge of their range, and who knows what they're going to do. Because as we move to the right side of these y-axes, we're having more of a novel climate. Whereas if we move higher on the y-axis or really low on the y-axis, we have more of a novel phenotype. And so it's not really clear under this counter gradient versus co-gradient scenario, like what is going to happen under climate change. On the counter gradient scenario on the right, some people have argued because northern genotypes have higher growth rates under warming, they might actually do better and have higher population growth under warming instead of do worse. But again, I'm not really clear if these predictions are going to pan out once we develop theory and actually test them. So lots of knowledge gaps. I think it's really interesting area, open with lots of questions right now. And I'd be happy to chat some more and answer your questions. Thank you so much.
Excellent. Thanks so much, Katie. Any questions?
Thank you. This is Stephen Gorin. I think this is really cool and interesting. In the, I guess it was the meta-analysis slide where you showed the scatter plot of everything. It looked like the Cove GE values were sort of reluctant to be around 1, -1, or 0. Do you think that that's something in your statistic that makes it like non-linear, or that that's
Yeah,
a biological signal?
No, it was because we only plotted histograms for the significant ones.
Okay.
And so we wanted to include a biological definition of significance in our analysis. And so we said if it's statistically significant and the effect size is greater than 0.1, then we would count it as, you know, a significant signal in our analysis. So that's why there's no points near 0. The reason there's probably no points near -1 or +1 is because those genotypes and their effects have to line up perfectly on a 1-to-1 line. So it's almost asking, like, why do we never see correlations of -1 or +1 in nature? It just doesn't happen. There's always random error in our sampling and stuff. Yeah.
Any other questions? I have one then.
Yeah.
So in your meta-analysis, do you think that could be in this question about whether co-gradient or counter-gradient variation is more common? Do you think there could be some detection bias in that the point you made about in counter-gradient variation might actually not be immediately obvious to us in nature? So would people have chosen to study that less than situations where there's co-gradient?
Yeah, and that's a really good point. Actually, maybe this is something Molly can speak to in the panel we're going to have soon. Is it was a lot of the studies that we drew from, like, weren't even thinking about gradient variation. They weren't even looking at it as part of their study. It was more the genetic bioenvironment interactions that they were focused on. And I think Molly actually quantified this in the data, and I'm not sure if we ended up plotting it, but it might be something interesting to include, is just like how many studies showed this gradient variation that weren't even looking for it. Now, the question you're asking is one from the perspective of, like, well, maybe if no one's even looking for it, it could be even more common than we're seeing in the meta-analysis. And that's one I can't think of the top of my head of, like, how would we begin to even know that? But if that was true, like, we would see much denser histogram on the left side of this graph than there is now. Yeah.
Any other questions? Or people in need of coffee?
Yeah.
Okay. Well, let's thank again all the speakers from these great working groups. And we'll have a coffee break now for the next 20 minutes, and then we'll start up again at 3:45 to talk about the future of the network.
Can you hear me on Zoom, guys?
Yep.
I can't see you, so give me an idea in the chat if you
Yes,
can hear
we
me.
can hear you.
We can hear you.
Okay, thanks. Okay. It's time to so we're gonna do the first of 3 discussions that we're gonna have about what the future of our network is gonna be. So to stimulate that discussion, they asked me to talk about how the network started and evolved through time. And I'm hopefully will give you enough information to give you enough background for this discussion. So Jeff Tressel brought us together. He was interested in starting a research coordination network for evolution in changing seas. And we put together a proposal that proposed to do a lot of the things that our working groups have been doing. And one of the reviews on our proposal said that we were an odd and inappropriate group. I don't know if you guys remember that. And then Dan Bolnick had the bright idea to like, let's take that and run with it. You know, maybe it's exactly an odd and inappropriate group to bring a fresh perspective to the field. And so you can always take something negative and turn it into something positive. Early on, Randall Hughes had a really important insight that to be inclusive, we should be an open network. So we made a decision to be an open, inclusive network. About 75% of our members are from the USA. We have 600 people that have signed up, but I think we have around 100 people who are like actively engaged in the network. And this is the distribution of how people characterize themselves. So the mode is in 50% evolution, 50% marine science, more skewed towards more marine science, less evolution. And that just might be a history of the field. And in terms of marine has been more focused on ecology in the past. We've already learned about a lot of the working groups. Later tomorrow, we'll hear from the Eco-Evolutionary and Oceanographic Models group. We'll hear from some of the Marine Omics website group, the Bioinformatics group, and Seascape Genomics of North Pacific forage fishes. So you've already learned about a lot of the intellectual merit that we've been doing through the talks today. So I'm not going to spend too much time on this, but these slides are online if you want to see. As some of the working groups have been talking about the importance of data, we do have a data page on our website, and data and meta-analysis data that is created through the RCN should be archived on BecoDemo. I know a lot of the groups now are not quite to the point where they're ready to archive your data, but just keep that in mind that we need to archive that data to be compliant with our NSF requirements for our award. And I think it would be great to add some more information to this page, like that we were talking about earlier, like the data agreements that some people have set up and other examples of maybe best practices for putting your metadata for your genomic data with the kinds of things that Rachel was talking about so that she can do the kind of analysis she wants to do without having to dig so much. We've also started an educational resources page on our website. So if you have a fun educational activity in your lab, we can just add it to the list. And we've just organized this by different grade levels so far. And it would be great to have more examples on this list. It doesn't have to be something from a working group. You don't have to be a PI. You can be an undergraduate or graduate student if you want to add something to Simulink. So we mentioned previously that we've done these coordinated readings every semester. And as you're thinking about the future of our network, I just want to share a little bit of data from these coordinated readings. So this is our Slack channel data. You can see that the bump every year has to do with the coordinated readings. They're every 2 weeks. So that's why we see these like multiple bumps every 2 weeks. The green line is the weekly active members. That's the larger line. And the blue line is the members who posted. And the coordinated readings have typically had about 100 people that have been engaged in them across a dozen different labs, posting their discussions on the Slack channel. Now, this last coordinated readings on temporal evolution, maybe people were getting burned out from the pandemic, but you can see that the rate of posting was going down and it was much lower towards the end. So I don't know if that's because people were not as interested in the coordinated readings, but the interesting thing is that the number of people active on the channel looking for posts was similar to previous years. And from talking to the junior people in my lab, they're really interested to see what's being posted, not so interested in posting themselves. So I think that that's something to think about. Is this, do we want to continue these coordinated readings? Are people burned out on them? How important, how formative have they been? At least I've heard a few people talking about how formative they've been for them. So it's something to think about in the future. So high rate of checking for postings, low rate of posting discussions. We've also had a lot of diversity, equity, and inclusion initiatives. Our diversity intern this last year, Janice Grisby, couldn't make it, but she's the one who designed our really cool, diverse RCN logo. And we've also featured our trainees, a lot of them who are here at this meeting, on our webpage and Twitter to highlight the diversity that's in our network and to help you raise your profiles as junior members of the network. You know, these are the kinds of things we need to think about. Do we going to continue doing these things in the future as the funding runs out to pay stipends for this kind of thing? We've also did a virtual lab meeting training program after the pandemic hit as a way to connect mentees, undergrads, grad students, technicians, in-betweeners with mentors. Mentees receive a $500 stipend for attending 10 lab meetings virtually and leading at least part of 2 of those lab meetings. It could be anything from leading a paper to asking questions to getting feedback on a manuscript. The mentors, we gave mentors and mentees a survey, and it's hard to see here, but this is the amount of time that mentors spent above and beyond their normal duties in this virtual lab meeting training program. About 80% of them spent less than 6 extra hours over 9 months because they're running their lab meetings anyway. So it's a great way to increase diversity, increase network connections without putting a lot of extra time on people who don't have a lot of extra time. The mentee statistics, mentees generally agreed or strongly agreed that the program helped extend their network, broaden their knowledge of evolutionary biology and marine science, and that they would recommend that program to a friend. The mentees also indicated that the ability to earn a stipend was pretty important to completing the program. So this is another thing to think about as we think about whether we want to continue these programs. How are we going to continue them if we want to continue them? That having funding to support stipends could be potentially like really critical to the success of the program. Mentees indicated that they were hopeful that in general their participation would lead to a future collaboration with their mentors. And so a lot of yeses, probably yes, might or might not. And then on a scale from 1 to 10, we asked mentors and mentees to rate the virtual lab meeting training program. And most of them rated it 8 or higher. So I think that on average, the average was around 8.5 for both mentees and mentors. In terms of critical feedback for this program, some of the critical feedback was from potential mentee-mentor matches who weren't matched very well. So maybe there was like, they weren't as interested in the mentors. And that made me wonder if we ran this again, you know, should we match people with someone who they're not asking to be matched with or not? But then I was talking to someone last night who was saying they were matched with someone that they didn't initially want to be matched with, or that they didn't, not that they didn't want to, but they didn't request to be matched with. And it ended up being like really formative for them. And they got a lot of knowledge about methods they can use for breeding and programming and R code, and that lab was sharing lots of information with them. So I think there's 2 sides to that coin. So what are the potential next steps for the network now that we're running to the end of our funding? And I've laid out basically 5 options from no work to lots of work for us. The first option is that we could end the network activities. We're all burned out from the pandemic. You know, we're all stressed really thin. We don't have a lot of extra time. Maybe this is an option that we should consider. The second option is that we could continue network activities unfunded on a volunteer basis. So this would be volunteers to continue developing some of the genomic tutorials that you're going to hear about tomorrow, building educational modules, running the coordinated readings, running the virtual lab meeting training program. We could talk about what initiatives we would prioritize. The third option is to explore other funding mechanisms. We could, for example,
submit a proposal to NSF for another workshop in a couple years. And we could decide what the objectives of that workshop would be and the program